This is the mitosis part 2 video, and we left off at prophase. So 3 important things happen at prophase. Let us draw our cell. Here's our cell membrane. Here is the nucleus. And we have our DNA as chromatin floating around. And now one organelle that we've briefly mentioned before, this is our centrosome. So this guy here is a centrosome. And this consists of those 2 small rectangles inside. Those are 2 centrioles. And I mentioned before that during synthesis, the centrosome doubles. So we have another set of these guys. And you'll see their role in mitosis in the next step. Okay, so during prophase, a couple things happens. This nuclear envelope starts to break down. I'll indicate that with this dotted line. And the DNA starts to condense into these chromosomes. And since synthesis already happened, they look like the X's now. And then now these centrosomes are going to migrate to opposite sides, to opposite poles of the cell. So this centrosome is going to come to this side, and then let's see, this centrosome is eventually going to migrate to this side. So 3 things. 1, the nuclear membrane breaks down. 2, the chromatin starts to condense to chromosomes. And 3, the centrosomes move to opposite sides of the cell. And this is mentioned up here. So that's prophase. Okay, now on to metaphase. So in metaphase, now that these centrosomes are at opposite sides, they release these projections that are made up of microtubules. Some projections go towards the cell side, some projections go all the way across, and some projections actually hook on to the chromosome, and they bring it to the center. Remember, there should be 46 chromosomes, but I'm not drawing all 46 because I don't have enough space for that. But you just know that you should have a line of 46 chromosomes. So in metaphase, projections bring these guys to the center. We also have some projections that go towards the cell membrane. Where they bring them, we call this, like this imaginary line here, we call this the equatorial plate. But this is pretty much it for metaphase. If you see chromosomes lined up in the center at the equatorial plate, you know that you are in metaphase. Oh, and these green projections, these are made up of microtubules. And then we call this structure here, we will call it it's called the spindle apparatus. We call it the spindle apparatus, just to give it a name, apparatus, spindle apparatus. Okay. So now let me take my diagram here and go on to the next phase, which is anaphase. Okay, so what happens in anaphase is that these microtubules are going to start to shorten, and they're going to rip apart all these chromosomes. So anaphase, here is my centrosome system. These microtubules, the ones that are hooked on to the chromosomes, are going to shorten. And as they do so, it's going to rip apart this chromosome. And now you can clearly see the second centromere. So I took half of this guy, I took half of that guy. The centrosome, the microtubule line that's connected to each other, nothing changes there, pretty much stays there. Here's my second combo system, and these also took part of that chromosome, exactly half. So now you can clearly see that second centrosome. And they're actually going to bring them all the way to each half of the cell. So these fibers shorten, and they bring apart the sister chromatids to each side. Okay, next is telophase. So after anaphase happened, we have our DNA in one area of each side. So here's what we're left with. Our centrosome system is pretty much done, but I'll just draw it in there because it's still there. Okay, so notice here that the chromosomes are kind of in the same region. So that's one thing you should take note of. So the chromosomes or chromatids, let's call them chromatids, kind of segregate themselves to one region, into one area. And that's because it's going to reform. We're going to have the reformation of the nuclear membrane. So the nuclear membrane reforms. So let me indicate that with this green dotted line. The nuclear membrane is going to reform. Second, the DNA is going to unravel and go back to its chromatin form. So chromatids are going to unravel to go back to its chromatin form. And then lastly, in the center of the cell, you're going to start to see a pinching in. You're going to have contraction of, you're going to have contraction here. And let's kind of take a snapshot of what we're looking at. So let me give myself some space. Okay, so there's my cell, but now we can start to see the beginnings of this pinching in. The nuclear membrane is reforming itself, and the DNA is starting to unwind, and it's going to go back to its chromatin form. And I guess our organelles are still there. And then same thing on this side. It's going to start to reform, and the DNA is going to start to unravel itself back to its chromatin form. And this is still going to keep on contracting, and eventually we're going to be left with two copies of the cell. So the nuclear membrane is back up. We can see our nucleolus, and we have DNA loose all over the place configuration. Let me draw that, nucleolus. Same thing on this side. Your nucleus is there, your DNA has finally unraveled. We can also still see, and the nucleolus also reforms. And you also have your other organelles in there. But here is it. The products here, we call these two daughter cells. Two daughter cells. Yep. Okay, so we did that for animal cells. Same thing for plants. One main difference is that they have this cell plate, which forms in the center. And they don't really have centrosomes. They have something else that creates the microtubules, but you don't have to know this for my class. Okay, let's take a few examples looking at some onion root cells. Okay, let me list my phases of the cell cycle. We have interphase, then we have prophase, metaphase, anaphase, telophase. Now I can see here we have a nucleolus, also there, also nucleolus, also there. All these cells that you can see their nucleolus, that tells me that it's still an interphase. So all these cells right here are examples of interphase. Okay, let's use a different color. This cell here, where the DNA is kind of at the center at the equatorial plate, I'm going to say this is metaphase. Okay, here I can see DNA start to being pulled apart and going to each side. This is clearly an anaphase. Okay. So now for an example of telophase. Okay, so telophase would just be, I guess we can call, let me erase this for a sec. If you look at this one carefully, we can kind of see this new cell plate forming here. So, and then this tells me that telophase is almost over, and that we have the reforming up of the starting material. So I would say this would be telophase. I almost got confused there. Okay, so to let me draw what this looks like on the side. So here's what we were just staring at. This is the cell plate. And then we can see our nucleoli, nucleoluses. And then we know that we have some DNA floating all over the place. Okay, so now for a second example. If this is the border of my cell and the DNA is at the center, at the equatorial plate, this tells me we are at metaphase. Okay, here is the border of my cell for this case. And I can see that the DNA is being pulled apart to each side. This tells me that we are in anaphase here. Okay, so all these stages up here, this looks like prophase because I can still see my nucleoluses. There's a couple there, prophase. Oh, sorry, not prophase. We can say these are interphase. We can say interphase, interphase. But once you start to see the DNA starting to condense itself, so along this direction, my DNA is condensing, we can kind of say this is prophase. So this one for sure would be a prophase. And this also looks like condensed DNA, so early prophase. Okay, here the DNA is kind of in one region. It's going to aggregate to reform the nuclear membrane. I will say this is early telophase. And this one here, we have our cell plate. Cell plate, the nuclear membrane is reforming, the DNA is uncondensing. This is late telophase. But that's it.